Hi, I'm Jonah Comstock, Editor-in-Chief at pharmaphorum. I'm here at JP Morgan. It's day 3 of the conference on Wednesday. I'm at the Hotel Sparrow, and I'm joined by Jeff Fischer, President of Longhorn Vaccines and Diagnostics.
Thank you for having me.
Yeah, thanks for speaking with me. So tell me a little bit about Longhorn. What do you guys do, and what sort of differentiates you in the market?
So we are a commercial diagnostic company that sells a product for sample collection, specifically around molecular testing. And then on the pharmaceutical side, we are a preclinical stage pharmaceutical company developing vaccines and monoclonal antibodies for the prevention of infectious diseases, as well as other inflammatory diseases that are caused by bacteria and other pathogens.
Why Longhorn? Are you guys Texas-based?
I went to the University of Texas for my MBA, so that's kind of where we pulled that name from.
Nice. Let's start with diagnostics, because I actually don't, I hear sometimes at shows like this, diagnostics is a little bit of the kind of black sheep of the of the industry. It's really important, especially as we move into a world of precision medicine, but I don't know, it can be less sexy and it can be less easy to see how you monetize it. So what's your philosophy when it comes to diagnostics, and where are you focused in, and how do you make that work as a business?
Well, accurate diagnosis is crucial to proper medicine, and as companies that are here are developing new drugs and new therapies, understanding what it is that you're treating and making sure that the medicines are well targeted is very important, because for a drug to be effective, it needs to be targeted to what it is meant to either prevent or cure. So highly accurate diagnostics really make drugs better. One example of that is we worked with a vaccine developer that was developing a vaccine for herpes. And prior to working with us, their placebo group was outperforming because their diagnostic wasn't picking up all of the infections. And so working with us, we were able to help them make their tests more sensitive. And so while their drug group still had fewer infections, their placebo group's infections went up, and that therefore made their drug clearly more effective. And so that was a clear case of their drug was doing what it was supposed to be doing, but they were not getting the separation because they weren't accurately testing their placebo group.
To be clear, you didn't make it so that more people in the placebo group got infections. You just made it so that the placebo group contained a different mix of people.
Well, so that we were accurately identifying. So they were missing infected people.
Right, and putting them in that group.
And so it was making it look like the placebo group was outperforming historical norms, which then requires a larger study and more money, and often can be the difference between that p-value being 0.05 or being 0.1. Where it had nothing to do with the quality of your drug. It just had to do with the fact that you weren't properly identifying the infections in the placebo group.
How did you end up doing being in the pharmaceutical side as well as the diagnostic side?
So we started this company back in 2006 after we had sold our previous pharmaceutical company after very successful phase 2 studies. And we wanted to go after pandemic influenza. And we realized that there were really two sides of it that needed to be improved to be prepared for what at the time we were concerned was going to be an H5N1 pandemic. One, we needed more accurate testing. We needed to identify the virus earlier and be able to make sure that we could potentially stop it where it was, or identify people early before they got sick. And so that's how we got into the diagnostic side. The other side of the business we wanted to get into was finding a vaccine, a universal influenza vaccine, so that we would be prepared when that virus became a pandemic and have a vaccine in place that could prevent people from getting either sick or dying from it.
Now, this is pre-COVID, right?
This is well before COVID.
Yeah.
This is 2006. This is even before the H1N1 pandemic. This was really early before people were really talking about influenza pandemics.
So when the H1N1 pandemic came along, were you well positioned at that point, or how did that affect your business? How did your business affect that?
We were. So our sample collection technology had already been developed. We'd already done a clinical trial in influenza patients, and we were able to demonstrate that using our sample collection technology and molecular testing, we could identify family members of children that were showing influenza symptoms but were asymptomatic 48 hours before they became symptomatic. So what we were able to do was really show the sensitivity of molecular testing could really identify people before they got sick. So when the H1N1 pandemic hit, we were in position to file an emergency use authorization, terminology
Yeah,
we're
yeah.
all very aware of now through the COVID pandemic. But during the H1N1 that was the first time that the FDA had really used that process in any kind of scale. And there were only 13 assays that were approved during that time. Most of them were government agencies filing with the FDA. So we were the 12th or 13th, which was good for us, other than the fact that by the time the FDA got around to approving the emergency authorization, the pandemic was over. And so, but it really led us to be prepared for and work with the FDA to get it fully authorized. And so when the when the SARS-CoV-2 pandemic hit, we were ready and set, and we were one of the largest producers of sample collection media during the SARS-CoV-2 pandemic.
So, flash forward to today, the world is more aware of pandemics, of the potential for pandemic, the impact of pandemics, than probably ever before in recent history. But at the same time, there's now all this kind of backlash against vaccines and all this misinformation, conspiracy theories. How do you? What are your kind of priorities in 2024? Thinking about the next pandemic. What are you working on, and how have recent events changed how you work, if at all?
I think what it's really taught us, and it really fits with what our philosophy has been all along, and that is that a really good pandemic vaccine needs to be safe, but it also needs to be effective. For people to really want to receive it, to trust it, it actually has to probably significantly prevent infections.
Which, yeah, the COVID vaccine didn't always do.
Right. I think that when we first started off, I wasn't sure we were going to continue as a vaccine company when the mRNA vaccines were showing 95% effectiveness for preventing infections, in the very first look. And we all found out later that they really didn't prevent infections. They prevented serious disease. They prevented some hospitalizations, probably prevented a lot of deaths. But they didn't necessarily prevent people getting sick or spreading it. And I think that the next phase for a pandemic vaccine, or for what we want to do with a universal vaccine that can be available before, is to have a vaccine that's truly effective. I've heard that this year's seasonal flu vaccine is about 25% effective. It's hard to convince people that healthy people that that's a good reason to go get a vaccine if it's only going to provide you a 25% chance of not being sick. So what we want, and we've been working on, is a very comprehensive approach to flu that we expect will significantly reduce that percentage year over year, and not rely on trying to chase the virus, which is obviously what we've been doing with SARS-CoV-2 and what we do every year with the influenza vaccine.
So I feel like I've heard this pitch a few times at JP Morgan from companies that are working on vaccines and influ and infectious disease. Why is it we think now we might be able to create something like you're describing, a universal vaccine? What's the science behind that, and what are the challenges to actually kind of, I guess it's a question of like, if we can do this, why haven't we done it yet?
Well, I think viruses, as we've seen with SARS-CoV-2, change all the time. And so finding a spot that we believe on the virus that is a consistent component that crosses all viruses seems like something we should be able to do, and we identify them all the time, but often then the virus finds a way to change. And so our approach has been to not pick one spot. What our technology has allowed us to do, both on the active vaccine side as well as the passive monoclonal antibody side, is to go after different stages of the virus's replication. So in our case, we target the hemagglutinin, which is how the virus attaches to the cell. And then we target the matrix component of the virus, which is how it replicates inside the cell. And then we target the neuraminidase, which is how it releases out of the cell. So our goal is to not pick one target, but to go after the three main components of how it continues to grow and replicate, so that it never has a period where it is not under pressure, and therefore it makes it a challenge for it to escape and modify itself. And so that's really been our approach. And so what we've been able to do is to create a peptide vaccine where we take pieces of each of those epitopes, string them together into a single peptide, put a T cell epitope on there to help adjuvant it, and then put an adjuvant in. And so right now we're pushing that through as an intranasal, which is a different to how we started. One of the things we learned from the SARS-CoV-2 pandemic is mucosal immunity may be the most important component to preventing the actual infection and spread. And so that's where we're taking this vaccine that we've been working on for 15 years, is now to see how we can generate that mucosal immunity. And we think that taking the approach we're taking and then going through the intranasal approach really may give us that opportunity to do what, as you've said, we've been trying to do, people have been trying to do for 30 years and have just not been successful.
The economics of vaccines and preventative health is always a little tricky because, you're in some sense you're trying to sell to everyone, but you're trying to sell them on the prospect of avoiding something that will happen in the future rather than something that's already happened to them. And if there's a pandemic on, that's an easier sell. But if there's not, it's a hard sell. How do you think about kind of when you have something in when it's approved and safe and effective, how do you get the word out? How do you capture enough market share, not only to make a profit, but also to make the vaccine effective, because it has to hit a certain amount of the population?
So we see really a two-tiered approach to that. First, we think that the fastest way to market and to demonstrate the success of this approach is through a monoclonal antibody cocktail, an extended half-life version of that, so that we can give that to people who are of high risk, people over maybe the age of 65, people in nursing homes, people that are maybe going through cancer treatments or other things where getting the flu could be really cost their life.
Yeah.
And the nice thing about that is you don't have to have the size, the safety database, because that's clearing in 6 months. And so antibodies have generally been found safe. The targets we're going after don't have any receptors on the human body that could cause other additional issues. I think we've seen with RSV, the Beyfortus, that monoclonal antibodies for viruses tend to be very safe. And so we see that as the first approach. And then really for that active vaccine, I think we have to be better. I think that's really what it comes down to. Even with as poor as the seasonal vaccines have been, they sell $6 to $10 billion worth of those vaccines every year. I think that if you could demonstrate that you're actually preventing infection, if you could get into the 65, 70, 80% reduction, I think people would love to have a vaccine that prevented the flu every year, if they could really trust it, and maybe not even have to get it every year. I think that if we do it right, it may be something you get every couple of years or maybe every 5 years. So we believe that that is the goal. It's not just having another vaccine that does what the seasonal vaccine does today. I agree. I don't think that that's a real good value proposition. But I think if you have one that is significantly better and doesn't chase the virus, but stands in front of the virus, I think that there's a tremendous market for it.
Yeah. And that said, we have the infrastructure already for those seasonal vaccines. We have the pharmacies, we have the insurers paying for the doses in many cases. So you don't have to make your pitch to every consumer, right? You have to make the pitch to those pharmacies and those insurers, to offer it and promote it, and you can get it out that way.
Which we feel great about because they're already covering a vaccine that's really not very effective.
Yeah.
So the bar's low, but we believe that to do what's proper, we don't want to just barely exceed that bar. We want to significantly move the bar up. and that's why we're really targeting 75 to 80% effective rate, and then maybe a multi-year coverage.
So where are you in this journey? What milestones do you have coming up?
So we just now started the approach of taking this vaccine, which we've manufactured into GMP, but we are now re-looking at it intranasally. As I said, we were ready to move it in. We had filed a pre-IND with the FDA, and we were close to going into clinical trials for an intramuscular. But now we really think that the right approach is intranasal. So we've taken a step back, and we're now going through the studies to confirm what we believe on the intranasal approach. And so assuming that those studies show what we expect them to show, then we'll start very aggressively moving through in 2025 getting that approach put into the clinic.
And yeah, when do you think it could be in the clinic by?
I think it could be in the clinic by probably the fourth quarter of this year if things move. The nice thing about it is we use a peptide approach, and peptides are very easy to manufacture. It's synthetic peptides. This is the same technology that's being used in the GLP-1s, but we're using a tiny fraction of the amount of material that they use. They're injecting large amounts of this every week into patients, whereas we're looking at very small amounts given maybe once a year or once every couple of years. So the technology and the scale has been done and really gone through the roof for these GLP-1s. So now we get to benefit from that. And so from a cost of goods standpoint, from a speed of manufacturing standpoint, we really get to benefit.
Sort of piggyback.
Exactly.
Yeah. Fascinating. Any other thoughts about the year to come? Especially, I've been asking people about the administration change, the regulatory headwinds. Anything you're nervous about? Anything you're looking forward to? Anything you're unsure about?
Well, so the other product that we're very excited about is we have a monoclonal antibody cocktail for sepsis that we've been working on. It goes back to the company that we sold in 2005, and some of the work we were doing then that we're no longer not allowed to work on. And so we're very excited about our antisepsis monoclonal antibody cocktail because we think it's going to have a significant impact on preventing sepsis, on antimicrobial resistance. But because we're targeting bacterial toxins as part of this cocktail, these toxins have been implicated in a wide range of inflammatory diseases. And so one of the things that we feel may be positive on the direction the administration is discussing is their goal of trying to reduce disease and trying to get ahead of disease. And while there's concerns about maybe some of their stances on vaccines, I think the idea of preventive therapy is something that I think that could be a positive in this new administration, as well as we have tremendous data already from previous clinical trials on some of our targets that these are safe approaches because we're going after cell wall components of bacteria. These are not targets that have any other benefit to the human body. So by going after something that's generally safe and we believe this could have significant positive impacts, there's been discussion about maybe being able to move things faster once they're determined to be safe and maybe not require some of the phase 3 studies that the FDA has required in the past. But we'll wait to see on that. But we're very excited. I think we're all uncertain about where things are going to be in the next 2 years. But I think generally we feel like there could be some positives out of it.
Thanks so much for chatting with me, Jeff. I really appreciate hearing a little bit about what you do.
Yeah, no, I appreciate the time. Thank you.